It's pretty self-explanatory. So all of us that start with a sequencing project with any type of genomic study have this idea that we have our creature and we have some very clear method how to sequence it, and then we get this beautiful telomere to telomere assembly with fully annotated genes, promoters, and repeats, so we don't have to like deal with all that stupid technical problems and rather do the real biology, right? But the reality is usually a lot sadder than that. We have this sequencing magic that is like generating us reads with the quality scores. We don't really know how exactly they are generated. And then we use assembly algorithms that we conceptually understand, but not really, because nobody really understands assemblies, because the assemblers have like hundreds of thousands of lines of codes and they're overly complicated. So, and then we have like thousands of unplaced scaffolds and all contexts and very fragmented genomes, and we don't really know what we've done wrong in our lives. The reality of it is that the sequencing is not yet there to give us telomere to telomere assemblies for every single genome that is out there, and therefore we somehow have to learn ways how to operate on this space of black magic and assembly magic. So this is kind of what this workshop is about. Like, we would like to discuss everything else than the assembly. So usually the genomic workshop starts with like, okay, we have the haploid reference and or like monoploid reference, and the read set, we map it on the reference. We would like to go a step back and just try to look more about on the sequencing process and demystify it, and give you a power in manipulating the sequencing data, not like to be able to do more than just map it to a reference. So I will start at the very, very, very beginning, just to be sure that we're all on the same boat. So we have all those creature without known genomes, and they have their cells, and in the cells they have usually the same karyotype, where in most common ploid level among animals diploid have each chromosome twice. Then these two chromosomes, which I conveniently drawn in the metaphase, can be then visualized as two linear haplotypes, because these are just copies, right? And when we sequence them, we do nothing else than we just crush this cellular matter, fragment it to pieces, and then read it, which as a consequence will just represent pieces of the two haplotypes we have sequenced. And if we would have a perfect haplotype and each read perfectly placed on its genomic position, then we could measure sequencing coverage, which would be the average number of reads supporting each genomic position. But in reality, we don't know the haplotypes. So what we actually have is this set of sequencing reads, and we need to somehow infer the haplotypes by assembling them, or haplotypes. We usually try to go for a single haplotype, so having like a monoploid reference. And the genome assembly is obviously fragmented and has its problems. The problem is that if we have a genome that has duplicated regions or very heterozygous regions, what can happen easily is that in the sequencing, in the assembly process, we will mistake these divergent sequences as two paralogous regions. It just might, what can happen is that these duplications we will just consider as a single region, which as a consequence can very often end up with hopefully monoploid references, but in fact kind of lots of representations with lots of problems. So for example, if we would map reads back to this kind of reference, we would see that the separated heterozygous regions will artificially lower the estimates of genetic diversity, because the purple and pink are divergent enough to attract reads from itself, like it's representing the two haplotypes, and therefore they will appear in the assembly as two completely homozygous regions. While the collapsed paralog, although being overall quite similar, might harbor a few slight differences, and though will then artificially increases the genetic diversity. And the biggest problem of it all is that we have very little power to detect these problems. They are really hard to quantify, because we just don't know. Like, we don't have a good measure for how much assembly errors are happening. So there are loads of techniques of how to stay sane while doing genome assemblies. Okay, I see that I'm getting questions, so that's fine, but I would ask other organizers to notify me when that's happening. Or maybe even just unmute yourself and jump in. So what I mean by collapsed paralog is that when we assemble a genome, we would like to have a single haploid reference. We would like to have it from telomere to telomere, but only one of them. And then we would like to map reads back and see that in this region they're heterozygous, and then we have these two paralogs that are in the reference separated, right? So when I mean collapsed, it means that if I assemble it as a single scaffold, not as two different scaffolds with a few differences. So that's what I mean, a collapsed paralog. Perfect. Yeah, I would encourage you to ask questions. I'm very happy to answer them. I might not notice them if you just ask me in chat, so please, yeah, you got the point. So the other sanity checks we're using are things like Busco, or in general locating conserved genes, because there was Sigma was called the one before, I think, which is just a predefined set of copies that we know they are conserved in clades. They are benchmark on universal single copy orthologs. So this is a set of genes that we know that we should find in nearly all genomes in a single copy, which is really nice metric because it sort of penalizes misassemblies. So if we have a pieces of genome put on different places and it cuts through the genes, then it will like lower the Busco. But the problem with Busco is that it saturates really quickly. That even very fragmented assemblies can have very high Buscos. So it does not have a sort of power to detect between all the assemblies. And it's, again, just a metric. It's not quantifying how the good, how many errors in the assemblies are of different types. The other very neat tool to deal with assemblies is blob tools, which is when we just look at the taxonomical assignments of individual scaffolds and plot the GC proportion against the coverage in the read set. And what you will end up with that you will see a few blobs that usually correspond to different sources of genomes. So this is a plot picture they have in the manual. So I'm not even sure which one is the core genome they were trying to sequence, but you see that this is sort of a metagenomic sample where you have this purple, red, blue, and orange genomes. So with this kind of plots, you can sort of stay sane by kind of considering also, wait, what if my library was not pure? And then in the end, the most powerful way how to look at problems is to also use other data. So when we're annotating, it's really neat to know the genome size using flow cytometry. Neat to have karyotypes. It's awesome to have optical maps or high-C data, which are just other techniques how to infer long-distance relationships in your genome. And when you're annotating your genomes, it's amazing to have transcriptors. But all this costs lots of money and lots of experimental effort. So this is basically the only part that is sort of assuming you have the assembly. The rest of the approaches we will be talking about are purely focused on Kmers. And for the part I will be mostly talking about is when Kmers are used to look at this pile of reads we are having. And the reason why I like it is because it really gives you insight about how sequencing works. And I hope that by the end of tomorrow, you will agree with me. So now we're finally getting to a Kmer. But what is a Kmer? So Kmer is just a substring. It's defined in a very general way. So it's any possible substring of length k within a biological sequence. The substring can be of a nucleotide sequence or a protein sequence. That doesn't really matter. But it just means that the sliding window, that we will read our original sequence in the sliding window type and read each possible Kmers. In this example, we have sequence of length 4 and the Kmer with k equal 3. So we have 2 Kmers in it. And yeah, Kmers are kind of everywhere. Like I'm 100% sure that all of you that have done at least a tiny bit of genomics have used a method that is using Kmers, because Kmers are really everywhere, and because they are not just a powerful tool for you, but they are also a very neat bioinformatics concept to store databases, etc. But we will get there. So I would like to just right now, before diving in lots of different Kmer methods, disclaim that this is my take, that the Kmer world is huge, and I have by no means expertise in all of the corners of the Kmer world. And as far as I know, I think this is the first workshop that's really attempting to like take Kmer as a concept and discuss it. So I did show you the sequencing process where we have the two original haplotypes that are represented with sequencing reads. And so basically counting the Kmers in your read set is something not that complicated. You have just, in this example, a read of 27 bases. Okay, I do admit this is a bit old-fashioned. These days you have usually more than 100 bases long reads. And in this example, the read is decomposed on all possible 8-mers. So the K is equals to 8 now, which means that a read of 27 bases will generate how many Kmers? Would somebody guess? So I try to use the thumbs up if I will say the number you think that many Kmers are in there. So 27, 26, 25, 24, 3, 22, 21, 20, 19, 18. Okay, it's 20. And this doesn't seem to be like the most efficient way how to ask questions. Sorry about that. And I hope that at this point you will appreciate how the logo of Oh No was constructed. That it's really and for disclaimer, it was other way around. I first made the logo and then I created this big chart. You see that everything is in there. Anyway,
someone is asking why 20.
Wait. Yes, sir. Sorry. So basically, if you would, so you would count the first 8 nucleotide from 1 to position 8. Then you would count the second 8-mer from position 2 to position 9. And now, which basically means that those first 7 nucleotides won't form their own Kmer because they are not 8 bases long. And the same is on the other side. So it's you ended up with 27 minus 8 plus 1 Kmers. That's like the number of Kmers you have in a read.
And Hannes had his hand up, so Hannes, unmute yourself.
I just wanted to say somebody had a question.
All right. Oh yeah. So you take each read, you decompose it to all possible 8-mers, and for every single unique 8-mer, you count how many times it occurred in the read set. So I made this like small example that imagine that the first Kmer is ACAGTCAT, and it occurred in total 13 times. Then you have the second, then etc., and you will have the whole database of Kmers. And what you can do is just to plot a histogram of these and see how many times which coverage you have seen. And this is what we call Kmer spectra. And what we then see here is certain sort of Kmer coverage, which is his mean in the dashed line, and its variance, symbolized with the arrows. And this is a concept we will go through many times, and it will be really the cornerstone of loads of these analyses. So properties of Kmers. K can be anything between 1 and the length of the sequence. If K will be equal to the length of sequence, there will be just 1 Kmer in each read or in each sequence. If the K will be equal to 1, then you will have that many Kmers in a sequence that is the number of elements in it. So if we will be talking about nucleotides, and since now on I will just talk about bases, Kmers and bases. So, but note that lots of these things are more general concepts. So for k equal 1, there are 4 possible Kmers: A, C, G, and T. And then if you increase k, the number of possible Kmers are increasing because number of possible combinations A's, C's, G's, and T's is going up exponentially. So it's always 4 powered by k. So we can calculate how many possible Kmers are there for lots of k's, and you say that it really goes high pretty quickly. So in all those k's for very low k's are always present in all genomes. But if you're reaching this like ballpark of teens, like late teens, you already have that many Kmers that not all of them will be present in each genome. So this is already in billions, and these are like even higher order numbers. And then if you have k's that are really high, like 80, 100, 150, you really have so many Kmers that most of them will represent a unique sequences. And the thing is, these different k's are used for very different bioinformatic problems. The larger k's are used for assembling genomes, the lower k's are used for characterization of sequences. And these middle k's are those that are, I guess, the most exploited, and they're used kind of for everything. So let's go over which k's are used where. So, but I also wanted to explain that actually when we're sequencing reads, we don't know if we have read the forward strand or reverse strand of a chromosome, because the sequences are not oriented in the sequencing machine. So as a consequence, we don't really know if we have sequenced a Kmer or its reverse complement. So what we usually do is we construct the canonical Kmers, which means that for every Kmer and its reverse complement, we count it as a single Kmer, which will then mean that AA is in practice the same as AT, AC is the same as GT, so always the reverse complements. So which means that the practical number of Kmers per K will not be 4 powered by K, but 4 powered by K divided by 2. And usually when we say canonical Kmers, we mean the alphabetically first, but it's not necessarily true. Maybe a very silly question. Sorry, we got another question. Is it interesting to use local Kmers, say from long read sequencing? We will be discussing a lot of properties of Kmers, so let's discuss this like a conceptual aspects later on. So actually it is possible, but we will get there. So let me go through the individual categories and sort of like give you an overview of what kind of methods are there. Like it's going to be very superficial because I tried to be brief. So the brown graphs are this like more very popular approach how to assemble short read data, and it just is an approach when you decompose a sequence, or like when you sequence with this kind of sequence with multiple reads, you construct a graph where all the edges are Kmers and then all the nodes are the K-1 mers. And then you're searching for what's called a Eulerian cycle or a Eulerian path, which is a problem where you're trying to visit each of the edges once. And this is quite computable. And this is a heavily efficient data structure, like a lot more efficient than just dealing with the reads. And which is something that just allowed us to analyze this huge data sets of Illumina reads that were generated for so many genomes. So basically, it's not like this Kmer approach would be in theory better than just aligning reads to each other and somehow finding a path between them. But this is just computable, and that's a huge advantage. And so the important bit is that the brown graph is just a data structure that allows us to represent very efficiently the whole read set at the same time. this representation is really useful. It's used on not just for assemblies, but also in probabilistic RNA quantification. So that's used by Callisto for sure, but I think Salmon as well, where the transcriptomes that are the reference are constructed as the Brown graph, and then the reads are probabilistically mapped on the Brown graph, and just and they estimate which is the most likely transcript that is represented by the read, and this is how they quantify the RNA-seq in a very efficient way. So you see that the application of the Brown graphs goes beyond just genome assembly. And this one interesting aspect is that if you're using the Brown graph, would it the two reads, for them to be consecutive, you need to they must be overlapping by at least K-1 nucleotides, because the neighboring nodes in the Brown graph are an implicit properties of the sequence. It's not like it just says, well, it's overlapping by 3 nucleotides, so they are neighboring. It's simply that always K-1 is the overlap between the two reads. So it is between two Kmers. And because all the reads are decomposed to Kmers and without memory of which read is which, they must be overlapping by at least K-1 nucleotides for them to be consecutive. So, oh yeah, sorry. What I wanted to also say that as a consequence, what can happen is that when you assemble reads with the Brown graph, it might happen that some of the contexts you will assemble will be actually shorter than the reads, which is actually quite silly. And if Jean-François Flo is around, he really likes to ramble about it a lot. And so now I would like to avoid the first rabbit hole, because what I told you about constructed the Brown graph with a single K is not what is usually done for modern Kmer, modern short read assemblers. The modern assemblers are using more than 1 K and consecutively increasing the K while remembering which read is where, which is a technique that is deployed by Space or BWise. And nearly all the modern short read assemblers are going in this technique of gradually increasing the Ks. But that's something that I wouldn't want to get into, just wanted to tell you that it does exist. And by the way, I will use these graphics for every single topic I will go very superficially on. So we discussed how haplotypes are sequenced, but the reality is that when we sequence reads, they do have errors. And the errors propagates, and the errors are with, okay, if we have a certain fraction of errors in our sequencing reads, they will be somehow distributed across our reads. And if we decompose the read into Kmers, then some of the Kmers will be still representing the correct genomic sequence, but some of them will contain the erroneous nucleotide. And as you see on this graphic, each single nucleotide error will affect Kmers and Kmers. And but at the same time, if you would think about what is the fraction of reads that is actually correct, it's a lot lower than the fraction of Kmers that are actually correct, because you see that even this read does have a single mistake, so it's not entirely correct. It generates about half of the Kmers that are right and half of the Kmers that are not. And because also the Kmers are the errors are random, it will happen that the errors are usually with a lot lower coverage than the genomic Kmers. So then if you will plot again the histogram of the Kmer coverages, you will see that the sequencing error Kmers will form this kind of tail on the left side, while the genomic Kmers will form sort of, again, a peak with its mean and a variance. And this is a very neat property of Kmers. This is actually one of the main reasons why we're going for Kmers, because we are able to kind of tell which of the Kmers are real genomic Kmers and which of the Kmers are just errors, which is a luxury we don't really have with the reads unless we get them already mapped to a referent. So now, this is this is sort of covering the really long, high complexity Kmers. Oh, okay. One thing I forgot to mention here is that if you will choose a longer K, a higher fraction of Kmers will have the error, because each error propagates to exactly K Kmers, which means that if you will choose a lower K, there will be a better signal to noise ratio. But it has other disadvantages, because if you go with lower K, they'll have a lower complexity, which means that the Kmers will become less unique in the genome.
Kamil,
Then,
you have only 2 questions on the chat now. So how do you know a base is erroneous?
So just assuming that the errors are random, we don't know which base is erroneous on the first glance, but we do know that if they are random, that they will have a lower coverage than the genomic Kmers. So basically what we can do is we can model it. We can model it as an error term by fitting the 2 distributions.
but that is also quite sensitive to the K you choose, right? Because if you choose a very high K, then a higher percentage of your Kmers will have the error, but also the error will more likely be unique. Whereas when you choose a very low K, then it might be still an error, but it might be the same as something that's actually in your genome.
Oh yeah, that's very correct. Yeah, so if you will choose K that is really, really small, it's true that actually with K number equal to 8, it's very likely that even if there is an error, the same 8-mer is still represented in the genome. So the peaks will merge for a completely different reason. Yeah, you're completely right. So, but over a certain level, which I would say is like 17, 18-mer. So if basically, if we're above this red line, there is a fair chance that an error will generate our genomic Kmer. If you are below the red line, it's less likely because there are so many possible Kmers.
Okay.
Kamil, you have 2 other questions. One
Can you
from
read them?
Yeah, sure. So Sabrina, she reads, sorry if I said your name wrong. This is the part I am most curious. It is not really intuitive. How could Kmer behave like that? Why is the correct on quotes, one normally distributed and the errors are in the lower tail?
Mm-hmm. So all this is based on the fact that the errors are rare. If the errors are not rare, then the distributions will be completely blended. But if the errors are rare and the depth is, let's say, sufficiently good, then basically the whole stack of the reads, or like the K, so now we have the haplotypes, and the stack of reads is representing exactly the same sequence. And because I will sample this Kmer 1, 2, 3, 4, 5 times in the visual example, but in reality it's usually a lot more than 6 times, then I will have a lot of the real correct Kmers because the errors are rare. And only a few of those positions will get the error and few error Kmers around it, which means that errors being rare will form a distribution around like with a lot lower mean, and as a consequence it will have this shape. So it's because it's Poisson distribution, right? It's like the rare sampling versus frequent sampling, and if we have enough samples, then we get something that looks normal. While if we have a few samples, we will just have this kind of shape that goes from steeply down.
I'm just trying to understand this the error detection as well, and so I'm just kind of wondering: is there an inherent property in different genomes where you only see certain Kmers? So if you took Kmer of 18, you just absolutely wouldn't see. You don't get a particularly even distribution of Kmers that can occur. Some of them are just impossible. Which is why the errors are rare. Is that kind of? Yes,
well, that's kind of the point. That well, not they are impossible, but they are just like there is a finite amount of real Kmers in the genome, which should be a lot lower than the number of possible Kmers.
how much does that vary based on organism, which Kmers can be present?
Yeah, that's a great question. So look at these numbers. So how many possible Kmers are there for different K? Basically, the genome size, the complete, like from beginning till end, is the maximum number of distinct Kmers that can be in the genome. And in reality, it's always lower because of the repetitive sequences.
Thank you.
So if we have like one of those absolutely enormous genomes like mistletoe, how many? Lucía, it's time to impress us.
Oh, how many?
How big is the mistletoe genome?
So it's between 85 and 100 Gs.
So it's about 100 gigs big genome. So that's about 4 times less than all possible 21 Kmers. And that would assume that every single Kmer in the mistletoe is different, and it's very likely not. So when we have the big genomes, they are usually very repetitive, right? So basically, when we get over the actually very small case, we're already getting in the complexity that is completely unmatched by real genomes. Does it make sense?
You have 2 more questions, 3 now. So Irene asks, the use of single and paired end reads will only affect the coverage?
Yes, practically. The paired end information is completely discarded when using K-mer approaches, usually, yeah.
And then what is your prediction about the graph if you are using nanopore sequence with an error that is known not to be random? By graph, I mean Kmer distribution graph.
Yeah, so I was thinking about it a lot. With in theory, if you would have infinite amount of coverage, even with Oxford nanopore, you would get a separation of what is real and what is not. Because the probability of the error is still random, and it will always generate more likely the real genomic Kmer than an error of a certain error. But it becomes a lot more sneaky. It's the Kmer methods are powerful for sequencing technologies that have a low error rate. Or
then the last question we have. Oh, sorry. Is there optimal Kmer numbers to be used for short reads or just for a random thing? How to decide that and why?
That's a great question. How to choose your K? I actually think I will leave that one unanswered because Lucía will talk about it a lot, about like how the choice of K will affect the shape of the spectra. And choose of K, well, that super depends on each individual method. So I was trying to show in these graphics that the different types of methods will require different complexities of Kmers. So if we're assembling a genome, we really want to have as many distinct Kmers in the genome as possible. So we really need to choose as high K as we have coverage for. While if we're seeding for mapping, just to speed up the process, then we need a lot lower K. Or if we're characterizing assembled sequences and just wanted to detect the systematic errors of polymerase, then we need to choose a very small K, like 4, 5, 6, 7. But anyway, so you have this distinct approach. Let's get at the end of the presentation. I think some of these questions might get answered inherently. So try to keep the questions to things that I said and it was not clear if I might ask for this, so we can get through it. What I was about to say. Well, okay, so now I will talk about things that I'm least comfortable with. So really cut me a bit of slack, please. So Kmers are quite used a lot for seeding, mapping, and searching for overlaps and classifying metagenomes, which is none of things I really do. But it's so when we sequence, so in Kraken they built a database where they had like a bunch of sequences they knew, and then they decomposed the query in all possible Kmers, and then they built a decision tree that classified them in a different phylogenetic relations. And on a different levels of classification, they have different taxonomic ranks, which is pretty neat. And they have also a different, like a Kmers that makes them unique. So like this is in this one first decision node, it's this one Kmer that decides whether it is one or another. And it's kind of like a fun approach to use Kmers when like to build a decision trees based on presence or absence of individual Kmers, and then just you can take like any meta sample or like newly sequenced sample and just get the set of the Kmers representing that sample through this decision tree and just figure out where it comes from. So this is the kind of how the Kraken works. And there are more approaches like this. There is another called Gotcha that works for like understanding of where the species come from by matching Kmers. And I also think that Siavash will later on talk about the other techniques, how to do this using even skimming data. Now we're avoiding a second rabbit hole. So there is also this very cool way how to efficiently bin Kmers, which is called minimizers, which is basically decomposing Kmers to sub-Kmers and finding the alphabetically minimal sub-Kmer of those Kmers. And this is something that is just using reduced representation and just allows like a fast matching of similar enough Kmers, so you don't have to go through the whole like Kmer space when you're searching for similar things. And this is really nicely described in this blog post by Titus Brown. So I heavily recommend to check it out. And also even the ways how to store Kmers, like how to build a database of Kmers, is a subject of huge research. And it was reviewed not that long time ago by Ryan Shiki and al. So I also recommend to see that. But it's a huge field, just simply how to store Kmers, not even like what to use them for.
There's a question about what does it mean to bin and hash?
Oh, sorry. So bin is somehow to store them in the categories that makes them similar to each other. And hash is to calculate a numerical value out of a sequence. So it's sort of like a hash is just a function. It's a function that takes a Kmer and gives you a number. And it can be whatever function you can imagine. And these hashes can be used to bin Kmers, to either like categories of similar or distinct Kmers, or whatever is the intention of the hash. But the hash is just a function.
It's maybe worth saying, Kamil, like the hashing is not specific to Kmers at all. This is just like a cryptographic technique that is used, for example, to store your passwords and stuff. It's just a way of essentially encrypting and reducing the information from any long string. But it's obviously particularly useful here.
Yeah, very true. So hash is like a very general concept. It's just a function that takes the strings and gives you a number. So Kmers are used as a speed up techniques for mapping. So because when we're mapping reads, we don't want only perfect matches. We want figure out if there is a heterozygous site, we still want it to be mapped to the same genomic location. Or if it's a different species, we would still know that they are homologous. However, if we would do a sort of like alignment of a read on every single position in the reference or in a reference database, it will take forever. So what is quite often used is Kmers for seeding, which is something that searches for exact matches of the query sequence, which is either read or a sequence, and just search for perfect matches within the database or within the genome, and then considering only those locations for building up the alignment. This is how BLAST works. This is even how BWA-MEM works, and we will actually exploit this property later on when we'll be doing perfect matching of Kmers to a genome. We will just tweak BWA-MEM by saying that we would like to have seeds in exactly the same length as the Kmers we would like to map. So this is, again, a Kmers where the concept itself does not give us anything other than a computational speedup, but it's absolutely essential because sometimes when we're working with these enormous data sets, doing it by the proper way, it's completely impossible to do. Like it's completely incomputable. And again, for overlaps of long reads, Kmers are used to like decide which of the reads are neighboring. So this is like one especially was useful at the very early stages of like long read technologies when the reads were extremely noisy and the mapping was really expensive. Then the first assemblers were using because then decomposing to very short Kmers would be impractical. So they were trying to assemble directly the reads, but searching with which the reads neighboring which was expensive, and basically using Kmers was a big step forward. So this is I think the least prepared part of the presentation, because calculating genetic distance using Kmers is conceptually quite simple, but practically quite complicated. So when you compute the Jacquard similarity, which is the intercept of two sets divided by their union. Yes, but then it sort of give you a value between 0 and 1 that gives you an idea about like how similar the samples are. And there are a bunch of approaches that are sort of taking two genomes and using Kmers. They're figuring out what's the union of the two. And there is, again, a big research about how to do this efficiently. These are all those mashes, sour mashes, like even approximating a phylogenetic distance using Kmers. And this is something that I would like to just mention conceptually, that it exists, because I really don't have a depth to give a proper lecture about it. And actually more rabbit holes I would like to avoid. And now that I think about it, I should even the last slide called the rabbit hole. You can use even Kmers for GWAS. There is this eLife paper that was published not a long time ago that was instead of associating genotypes with phenotypes, as normally is done in GWAS, it's directly associating Kmers with phenotypes. So sort of like avoiding the snip calling step, or like using even the panels. And while I was preparing this workshop, I also found that there is this kind of GWAS type of approach applied for sex chromosomes, which I actually never seen before. So there's something that potentially is automating what we were doing kind of manually in our lecture. And there is also a concept of gapped Kmers. I feel Lucía will mention it a tiny bit, but you can also not do a Kmers as a consecutive strings, but as strings that have a gap in between. But that's again a concept that is unimportant for the basic usage of Kmers. So now, as the last bit of this introductory lecture, I will be talking about this very low case, because I actually a long time ago did quite a bit of research on that. And I think it's actually the most fun. I would say the least useful, but the most fun part of Kmers. Let's play a game. This is called a cow's game. When you take some sort of shape and label corners with labels. So in this case, in A's, T's, G's, and C's, but it can be whatever. And then somehow generate either completely random or pseudo-random numbers and start drawing points that will be halfway between the last point and the point you roll. So for example, in this case, so you always start in the very center, and in the sequence A, T, C, C, I would draw a new point halfway between A and the center. So that's over here. Then the next point is T. So the next nucleotide is T. So I do it between of last point, which is the only A, and place between A and the corner that is labeled as T. And then I have C, C, so I go C and C. So this is just a game. And what is it for? Well, it's actually really bizarre, but if you do it for like 15,000 nucleotides, so if you take like a random pieces of human genome or Arabidopsis genome and do these pictures, they're remarkably similar within the genome and remarkably distinct between genomes. So these are the randomly selected 15 kb windows in Homo sapiens, Arabidopsis thaliana, and E. coli, and look how different images they are, but how similar they are within the genome. And by the way, these images are called Chaos game representation, and it's just a phenomenon that is actually not super well understood. And the way it's used quite often is to, well, okay, and so how does it relate to Kmers? Like why I'm even talking about something that's called Chaos game representation? Well, the thing is, this is just a way how to create a picture of Kmers. If you would cut the image by a certain amount of evenly spaced lines, you will actually get Kmers of a certain size, and that the size depends on like how finely you cut the image. So in this one example, when we cut it by 8 times 8 lines, we're getting all the possible trinucleotides if we sum up all the points that are in the squares that you see. Because every time, regardless of where you start, if you roll 3 Gs in the row, you will always end up in this square. And again, regardless of where is the last point, if you roll TGG, you will always end up in somewhere within this square. And so this is sort of like a fun way to visualize Kmers. There is not actually a reason. If you would like to use a trimers, making this image and then cutting it, it's a lot slower than just counting the trimers. But it's just a very pretty thing to visualize first. And second, it gives you any K. So it's sort of, the image itself contains a full information about basically the whole, all possible Kmers that were in the sequence. And the remarkable similarity is actually something people study. So it's quite often, so people were exploring of how different it is between species. So these are like 5 eukaryotic and 5 prokaryotic genomes, and just a PCA of tetranucleotides of 5,000 windows of genomes decomposed to 5,000 base pair windows. And if you just plot the first 2 axes of the PCA, you see that the genomes vastly cluster with themselves. So just by taking a random 5,000 nucleotides and decomposing it on all possible tetramers and looking at their frequency, you can kind of guess what species you're looking at, or at least in which ballpark you are. And furthermore, it's not just the species that you can distinguish with it. If you cut a genome to different genomic features like coding regions, introns, low complexity regions, RNAs, tandem repeats, whatever. So this is a C. elegans genome, and then you plot the windows with the individual features again on a PCA. Then you see that again, even if features are clustering together. So the separation is not as nice as in the between species, but it seems that even the different genomic features have their own unique signatures. Which is, again, this is an interesting phenomena. It's not so widely used, not even that widely known, but I just but sorry.
Kamil, have you ever seen these being used for species complexes or very close related species, like we saw in the presentation yesterday for the Anopheles? Just curious.
Oh yeah, I tried to, well, species complexes. I tried to human and mouse, and already human and mouse are a lot harder to distinguish. So one of the reasons why these are so nicely separated is because they are all phylogenetically really distant species. So the species that are more closely related have a lot more similar genomic signatures. So I think for species complexes like Anopheles, you would need to increase the K and start to look at the unique case for individual lineages rather than like looking at Kmer frequencies. But actually, I'm not 100% sure because nobody ever tried.
Yeah, that's cool. Thank you. Cool. Very cool thing.
Yeah. So what is it actually used for? So it's quite popular among detecting candidates for the horizontal gene transfer. I actually authored one of the methods. It's not the very best one. I don't recommend people to use it, but this is the reason why I'm excited about it. That's why I know about it. So basically, the idea of these horizontal gene transfer methods is that it's scanning this genomic signature alongside the genome, and finding regions where it's really deviant to the rest of the genome, which is sort of like a signature of being origin from a different species. It's also used in this Anvio tool which is sort of like a metagenomic contamination analysis tool. It's also used in phylogenetics, but I vastly read just papers that are sort of talking about this as a concept. I could not find a paper that would present a software that would just simply allow you to calculate genetic distance from genomic signature, but it should be theoretically possible. I found papers that are defining it as a concept. So it's just, the genomic signature is just fun. The reason why I mention it is mostly because it's fun. So we covered the low case, the very low complexity Kmers that we definitely see in all sequences. We covered high case, Kmers that are with a very high complexity. We a tiny bit brushed some of the middle case, but the biggest use, or at least the one that we are most interested when we're looking at non-model organisms, are descriptors of sequencing libraries, because that's something where it's not inside of a tool. Like when we're blasting, we don't actually really care that blast is inside using the tool. What we need to understand is that it's somehow approximating the homology in the database. But when we're characterizing sequencing libraries, it's essential to understand how the Kmers work and what are their properties. And this is what the rest of the workshop will be about. So basically, now the fun part will start. So I will ask, do you have any question for any of the non-characterizing sequencing libraries questions?
So, hi, Kamil. Yeah, can you hear me?
I can hear you.
Yeah, thank you. I do have a question, but I think it's not very deep, and it's something you mentioned just very shallowly on this of separating species using Kmer frequencies and that is not used for that. But anyway, you say that for more closely related species, you could increase the Kmer size?
I guess, like for different cases, you're looking at different things. If you're like reducing case to very low K, what you're actually detecting are very subtle things like the systematic errors of a polymerase. Well, at least this is so far the best explanation of why the genomic signature is species specific. Well, if you start increasing case, you will start to have Kmers that are unique to certain sublineages, and like basically longer K. So if you're looking at these complexes that are very closely related, it's very unlikely that there will be a big changes in the frequencies of short Kmers. But there might be some already unique Kmers specific to the lineages. So that's why I was thinking that if I would be looking at the species complex of mosquitoes, I would probably increase the K a bit.
Yeah. Okay. Yeah, I just wanted to ask because for me that was a bit counterintuitive, I would say, because you could find more differences, I guess, just comparing smaller pieces of K, I would say. So that was a bit, that's that
Sure.
was. Yeah.
And also, like I try to cover the two extremes, like very low case and a very high case. And for those middle ranged case that are used in the mapping, I just try to conceptually describe how it works. But Lucía will right now give you a bit more explanation of how the K is affecting the Kmer spectra and like what within the genome profiling or characterizing genomes, how the Kmer length affects that.
Okay. Yeah. Thank you. Yeah.
Do we have more questions?
So if we don't, I would probably give it a break for 10 minutes or 15.
Yeah, that sounds good. 15 minutes.
Okay.
So the question is regarding.
Oh, go ahead.
Regarding Kmers with sequencing data with non-random error, you answered the question indicating that the error actually was random anyways, but it's not. can you expand and clarify your answer? Yes, sure. So when we say that errors are non-random, we don't really mean that they are not random. What we usually mean is they're not uniform. That practically means that if we have certain types of sites, they're more likely to have error of certain kind than of different. For example, if you have a CG site, it's more likely to be read as CC site than a CTE site. And like there can be like lots of chemical reasons for that. But the errors still occur by chance. So regardless of errors not being really uniform, the only way how to make the only reason why the increased coverage would not help us to separate the peaks would be if probability of a particular error would be same or greater than having the real genomic sequence. So it's just like sequencing error is not really so we would like to have a fixed probability that an error that a base is misread as something else, and we would like it to be regardless of what we are reading and the error that we're getting is also random. This is the ideal case, but this is not the case. So by non-uniform, I mean that for different base sequences, we have different probabilities of them being read as a different nucleotide. And it also applies for indels, that different sizes of indels have different chances. And so this is what I mean, non-uniform. And basically, like different technologies have different distances from uniformity. But while Illumina is quite good in the error profile itself, it's not perfect. That they have like the coverage bias towards GC side. Then we will have but they almost don't have any indels, while the Oxford Nanopore has lots of indel problems. So it's, again, a completely different error profile. So perhaps we should not ask if it's a random or not, but what's the distribution that drives the random?
Then there's another question, Kamil. Can you be more specific to how it is non-random? Do you mean the homopolymer and indels? Maybe we can figure it out together.
Yeah, I hope that.
I'm sorry, that's a reply to the other question, so we could figure out together if.
Oh, sorry.
Kamil already handled that, so sorry.
Yeah, I can't really see participants, so I'm not sure how many are back and
54.
whether it's time to start.
54 people.
Yeah, it's almost 20 past. I think we can start.
Cool. Okay, so yeah, it's time to introduce everyone to the K-mer spectra. I know Kamil has been talking quite a bit about them, but what I'm planning to do with him in the next couple of hours is basically like take you step by step on how the K-mer spectra is built, so it makes sense. I basically focus this presentation in a way that I would have liked someone to explain it to me on the first year of my PhD. So I was getting a bunch of genomic data and I was building these graphs and I had no idea what they meant for my species. Basically, that's the whole point of this. It might go back to some of the concepts we've just talked about with Kamil, but I think it's also good to sort of, bring things back and share some concepts. Oh gosh, I can't. Okay. So yeah, K-mer spectra, as we've already heard from Kamil, what K-mers are, we we know already. There are substrings of length that of K that contain a biological sequence. And we know that sequencing errors affect a smaller fraction of Kmers compared to reads. So he already talked about this as well. This is also something we've already mentioned. So how many Kmers are in reads? So if we, the possible Kmers that we get are depending on the length of the Kmer. So we know that we have 4 different AGTC, and then the different amount of potential different Kmers we get are dependent on the length, but the amount of Kmers in reads will decrease with the length of the read. Sorry, with the length of the Kmer. So the number of reads, the number of Kmers in reads will be the read length minus K plus 1. We've also mentioned that, but I don't think we showed the formula. I mean, it's pretty simple. So the Kmer power decreases with the Kmer length. This is another of those rabbit holes that Kamil was talking about earlier. Just wanted to talk about the concept of space Kmers. We've been saying that the amount of Kmers within a read, it comes from the Kmers going one after the other, like shifting from one base to another base pair. But there's also a concept of space Kmers is based on patterns of deterministic positions instead of contiguous Kmers. So this is used for some other techniques, like in this paper of alignment-free sequence comparison of space Kmers. I had a look at the paper and it looked really interesting for some specific methods, but we didn't really go much deeper. We just wanted to let you know that we can use Kmers in a different way that are not using all the possible Kmers in a specific read. So what we are gonna show now is basically how we obtain the Kmer spectra out of our read data sets, right? So if we had our read here, we've already said we will be selecting our K or our Kmer length, and then we'll be taking the amount of base pairs starting from the beginning, and then shifting one after the other. And then this will be our Kmers. And then after we get all the different possible Kmers we have on our data set, we will count them. And that will be our coverage. So how many copies of this specific Kmer I have in my data set. And that's what we are going to be calling the coverage. So when you plot your Kmer spectra on your Kmer or your Kmer profile, you are making the y-axis the number of unique Kmers. So basically, how many of these unique sequences I have on my data set, that have a specific coverage. So in the x-axis, you're showing what coverage every unique Kmer has. So we've already gone through this. So this is what goes from haplotype to sequence reads to Kmers. And then, how you count each Kmer from each read. So we'd start from my hypothetical perfect genome where all the sequences are in the same sort of amount. We don't have sequencing bias, we don't have sequencing errors, and every sequence, every Kmer is shown in a probabilistically in the same amount. And in that case, we would have basically a normal distribution of the different Kmers, and we would call that the main Kmer peak. However, we know that sequencing has errors, and we've talked about this as well, and how the different random sequencing errors can affect our Kmers and the specific amount of Kmers. So the one sequencing error on a read can affect a number of Kmers, but then the count of those Kmers, because they are random, it's very low. So that's why on our spectra, they will just show on a very low coverage right next to 0. But they will be in very high amounts. And then we also will have repeats, right? So repeats will be regions that are in a much higher amount. And therefore their counts will be much higher, and they will be showing the last tail of our spectra. So depending on how repetitive your genome is, you'll have a little bump at the end of your spectra. So we've also been talking about how much K or your Kmer length can affect your spectra. So basically, we know that the longer the Kmer, the more different possibilities or combinations there are, right? We've been talking about this. So the more different unique Kmers there will be, so that will affect the height of our Kmer peak. And we also know that the more different Kmers, the less copies of each there will be. Also because of the property that we've been talking about, that the longer the Kmer, the less Kmers there is in a read. So the coverage will be going down. So if you increase the K, your curve will be moving and approaching to the error asymptote. And moreover, the more of different unique Kmers, we've also been observed that the more accurate the coverage. So the profile will sort of be narrower the higher the K. So this is just for you to see how it varies within one single spectra or species. But something to take into account is this is the behavior or the Kmer spectra of something that shows one single peak, which means that there's the same number of copies of everything, meaning that this is probably a highly homozygous or a haploid individual. So there's no different alleles that will be showing at different frequencies. But that's not the case for most of the species that we work with, right? We also have different haplotypes. And that means that in some cases, we will have the presence of different SNPs or variants that will show up in half the Kmers, but not in the other half. And that means that from a pretty haploid peak, we can get our monoploid peak, which will have half the copies or half the coverage of our main peak. And this includes all the heterozygous Kmers. So the problem with having a sequence that heterozygous genome is that, of course, in broader terms, the more heterozygous my genome is, I will have a more fragmented assembly because my Kmers will be split into the two different haplotypes, and it will be fairly difficult. It depends on the type of data you have, of course, and there are many different methods to separate haplotypes now. But if you only have short-read data, it can get tricky. And the problem is when you have these heterozygous Kmers, you have what is called bubbles on the Bruin graph, in which, as you can see here, these different haplotypes have generated two different possibilities in your graph, and that can be difficult to resolve sometimes. However, as Kamil was saying as well earlier, there are very specific assemblers nowadays for everything. And there are some of them designed specifically to deal with highly heterozygous genomes. So, it's not the end of the world. This is just an example to show you that heterozygosity can get really high. And in some cases, the heterozygous peak can be even bigger than the homozygous peak, than the homozygous peak. And this is just actually a curve from one of the species from my PhD that I sort of ended up concluding that it could have been a hybrid specimen from the garden. So the botanics, we deal with a lot of horticultural varieties as well. And we thought this was something collected from the wild, but in the end, you don't know who has been playing with your sections. So they were actually hybrid specimens. From your Kmer spectra, you can get a genome size estimate. And that will be very, very similar to our to the coverage formula. So your genome size will be basically the total number of Kmers, but you will want to remove all the estimated Kmers with errors, and then you want to multiply it by Kmer length divided by the peak coverage times 2, assuming we have a diploid genome. However, this is sort of intuitive, but because of many reasons that we'll talk about later, sometimes it's much better and very important to have some idea of the flow cytometry data from your species, so you sort of have a clue that your data is in the right ballpark. Sometimes you might have sequencing biases, or you may have be missing like some super repetitive Kmers that are outside of your spectra. So there are specific cases where you might want to keep an eye on your flow cytometry data. So what types of data can I use to get a Kmer profile of my genome? Ideally, what we want is high coverage data sets. We want as many copies of your Kmers as you can, of hopefully Illumina reads from PCR-free libraries. This is the ideal case scenario. When we talk about high coverage, the problem is if you don't have enough coverage, you can't sort of separate your error Kmers from your low coverage Kmers. So here, this is the case of another of my species from my PhD. And as you can see here, the heterozygous peak just overlaps with the error peak. So this can be a problem with your assembly. And this is just an extreme example of mistletoe. We're slowly sequencing mistletoe at the moment, but of course, because of the humongous genome size, at the moment, the coverage we have is so low that it's just one error asymptote. So everything is together. We talk about Illumina reads as the ideal ones because they have the smaller error rates, as we know. But you can also use long read data, high C or 10x. I know we've been already discussing about whether a nanopore has random errors or not. I haven't really used or drawn Kmer spectra out of nanopore data, so I don't really know how the quote non-error random errors unquote can affect the spectra. But because they have different error rates, I wouldn't recommend combining the data sets and to making the one Kmer spectra, because I considered that with mistletoe because we have such low coverage. I thought maybe if I combine all the data sets, I'll actually get a spectra, but it's not. I don't think it's a good idea. And yeah, we just want to mention that PCR-free is ideal, but library because it's more bias-free. But we all work with non-model organisms, and we know sometimes it's just not possible to get PCR-free libraries. This is just to show you a bit about this really cool resource from the Tree of Life. This is called TalkQC, and they upload there all the Kmer spectra from the species that are being sequenced. So you have a data set, you can look for your species of interest or a specific taxonomic group, and you can see what kind of data they're generating, and you can see the Kmer spectra they've been drawing with all the data. So for example, for this species, they have uploaded, I think they also have high C spectra on the database. I just added this street for comparison of Illumina and PacBio. I did find striking how Illumina and PacBio spectra are so similar, because hi-fi data has very low error rates, and it's quite interesting. But we were talking about this with Kamil yesterday, and he pointed out, I didn't notice, but actually you need much lower coverage of Illumina to get a proper Kmer spectra compared to PacBio hi-fi. As you can see, the coverage of the main peak is much higher. Well, not much higher, slightly higher. And then you can also get your Kmer spectra with 10x, which is slightly different, but it shows more or less the same properties. And of course, you probably need more coverage for that.
Lucía?
Yes? Shall I look at the questions?
Yeah, so do you want me to read them or do you want to read them yourself? Yeah, you can read them.
Yeah, sorry. I don't know where they start from for me. Is it the Ronald question? Or...
Yeah, it's Ronald.
Yes, sorry. It's a spectrum, the Kmer coverage histogram of just a particular K and spectra. Yeah, so spectrum is the singular of spectra. Sorry if I've been saying that wrong. It might be. Oh, what's a low and high coverage for an organism if it's quite subjective? Any comments on that, and how do we decide? I would say it's very different depending on the organism or the genome characteristics and the sequencing technology you're going to be using. As a rule of thumb, the tree of life, which is sequencing every eukaryotic species in the British Islands, so it gives you sort of an overview of everything. They're using 30 times per haplotype on hi-fi data, for example. So that's more or less what you need for hi-fi data. But I would say for nanopod, you would need much longer coverage because of the higher error rate. And for Illumina, I would say as well. So yeah, it depends on of course, your read length and the properties of your reads. So yeah, okay. So we've gotten to the point that we're just stopping listening to me, and we're gonna get on first hands-on exercise. So we're gonna be making a first Kmer profile, and we just need a read file. And there's plenty of Kmer counters out there, but we're gonna go with KMC for the practical. It's Kamil's favorite, so it was his choice. And then just choose a K and run the counter and plot your results. So the specific instructions are on the GitHub tutorial. And the one thing that I wanted to point at is that if you didn't do the batch refresher yesterday, you will need to do like a previous step, I think, to allow the Conda environments on your account. So if you could just go back to the batch refresher wiki and follow those steps, then you can follow the introduction on Kmer spectral analysis to do the KMC count. And we say 20, but we are pretty flexible with that, I guess. Or do we want to meet at o'clock? What do you think? Let's say 20, and then if we need more time, we'll just say it here.
Yeah, so it's just we'll meet again at 1 o'clock, so in 20 minutes.
Okay, let's meet again, and then if someone needs more time, we can.
But I would suggest you to do it in groups. Jose, could you create?
Had a good first experience on building your Kmer spectra, if it was your first one. I'm just gonna continue talking about genome characterization. Yes. Can everybody see the right screen?
Yes.
Everything is good.
Thank you. Cool. Okay, so the second part of today's blog is how we can use GenomeScope to fit best Kmer models. Probably the right person to talk about GenomeScope will be Kamil, but he kindly handed this to me. So basically, from the problem that from a few years till now, many known model organisms are being sequenced, and we don't really understand or fully understand the genome characterizations that we are facing. or different haploidy levels or heterozygosity levels, or like super repetitive regions that we don't know how to handle. So Kmers and characterization of genomes using Kmers are a good way of sort of realizing of all these flags that your genome should have before the assembly. So what the GenomeScope designers did was basically design a bunch of statistical models for genomes with a specific genome sizes, heterozygosity, haploidy levels, and repeat content. And the basis of GenomeScope is you upload your Kmer spectra. You will generated a histogram file just now. So you use that histogram file, and GenomeScope will try to fit it into one of these many different possible models, and will tell you how close your data set is to a specific model that has a specific heterozygosity level, a specific repeat content, and a specific genome size. How GenomeScope works. So there's actually a web-based tool that is in the link below. We can share it later if you want. I really like the online tool. For me, because when I started my PhD, I was really, really scared of running things in the command line, and that helped. But also, and that's not the only reason, it's because it includes a few examples that are very illustrative if you haven't seen a lot of Kmer spectra when you're handling your own data. So if you go to the examples bit, they have different different species from very different taxa that show very various levels of heterozygosity. And I think they're very interesting to just have an overview. And of course, you can use GenomeScope on the command line. That's where we're going to use it. And it's very easy to download and install also from the GitHub repository. As I said, what you input is just basically your histogram file, which is a text file or a table with the Kmer coverage on the first column, and the second column is the number of coverage, and then the second column is the number of unique Kmers with that coverage. And that's basically what you've also used to draw your histogram. And then the output is something like this, which I think is really cool. So basically, I'm showing here one of the examples that I said the online version has. This is an F1 from 2 different accessions of Arabidopsis. And that's why we see a pretty heterozygous peak here. And basically, the main outputs are your Kmer curve, which is, so your data is the histogram is the blue bars, and then the full model, so the model that GenomeScope has adjusted to your data, is the black line. So you can see already from the spectra that it fits quite well on your model. The errors are the fitted to the orange line, and then what you can see from here is sorry, I guess you can see what I'm pointing at, but basically the right spectrum is just a zoom out of the first one. So you can see here, well, in a logarithmic scale, but what you can see here is the same spectra just zoomed out. And so, as you can see here, the different peaks of really, really high coverage, which I think is really cool too, and where your model is fitting inside of that. And then it also outputs a few text files, and one of them is basically your main results. So with 21 Kmers, the different properties of your genome as fit into this model. So have quite high heterozygosity, the length of the haploid genome, then the repeat length with the unique sequence length of your genome, so the non-repetitive regions, and then the duplication level of your reads. So this is useful for PCR, non-PCR free libraries, I guess, and the read error rate. And then here it gives you also the model that your data fits into. So yeah, so what things do you need to know when you're fitting your model into GenomeScope? This is just a few sort of tips or some advice when you're using GenomeScope, because it's very tempting to just get your data and do input it into GenomeScope and just getting some data out, but you actually have to be careful to check that your model is actually fitting well in your data. And the first thing you have to take into account is that you have to get your 1N coverage right. So this is an example of strawberry data. And what you can see here is that you can get, for example, if you run GenomeScope with default parameters, this little heterozygosity peak is included into the error line or into the error fraction of the data, and it takes your homozygous peak as a heterozygous peak. So in this case, this would be considered just a highly heterozygous diploid strawberry. While actually, if you specify a specific parameter on where your monoploid peak should be, it will actually fit much better on your model when you tell it, okay, this is actually my my heterozygous peak, and this is my homozygous peak, and this is my repeat tail, and then you can actually tell that your model is much better fit. So you have to be very careful with this kind of things, and you have to tweak sometimes the parameters for that. And then you can also, you have to also sort of take into account that you can input the ploidy level of your organism when you use GenomeScope. So having a previous idea of what the ploidy level of your organism is quite helpful. And also having a sort of range on where your genome should sit on genome size spectrum. It's also helpful. So just not to trust blindly what GenomeScope tells you. So, because for example, in this Saccharomyces data set, what we also have is it's taken, as I said earlier, it's taken the first monoploid peak as an error distribution, some kind of error. And then it's taken this as a diploid organism in which you have here one heterozygous peak and one homozygous peak here. But as we said earlier, if you fit your model correctly, you actually realize, oh, wait, this could be another haplotype. And then you have 3 peaks here. And that can tell you, actually, maybe this is a triploid organism. So if knowing the ploidy level of your organism could be quite essential also to fit your model. Again, here, another rotifer example. Kamil, I'm talking about your example. So if you want to comment on anything, feel free to say.
The one that said the Saccharomyces was tetraploid, not triploid.
Oh, yeah. Sorry, I missed that. Did I count wrong? But where's the? Okay.
So usually when you have a tetraploid, you still have just 3 peaks. You have usually peak that is diploid, the peak that is haploid, and peak that is tetraploid, because the triploid peaks are usually absent in tetraploid species.
Right. Oh, okay. I don't think I've seen that many tetraploids then. Thank you. And again, another example of a rotifer. And yeah, it's a very similar case in which if we don't fit the error peaks well, we can, and they're very low coverage, like the monoploid peaks will be very low coverage. So they might get absorbed by the errors again. And we miss one of the peaks or one of the haploid genomes. So, so yeah. Do we actually know what the correct answer is for this one, Kamil?
This one is absolutely bizarre case. So we
okay.
We noted that the total genome size is supposed to be about 250K. 250 megs. So that means that if it would be diploid with 90 is too small, and if it would be tetraploid with 90 is by far too big. But the thing is that even the tetraploid model on left does not explain fully the Kmer spectra. The best fit would be that the coverage would be half of that. And then we would have like 800 megs genome in total, which is just completely off what flow cytometry tells us. So this is like one of those really bizarre cases where we actually really don't know at all, because the sequencing of this specimen resulted in a completely different answer than the flow cytometry.
Okay, so this is basically what we don't want to find in our data.
Yeah, and we have a question now. What's the aneuploid? What happens to aneuploid cases? So would you like to get the answer now, or would you be patient for later on? Because we do have a bunch of Kmer spectra that are from mixed tissues of different karyotypes, and it's quite fun. You're right. It creates peaks that are unevenly spaced. And basically, okay, perfect. So we will talk about it later.
Cool. Yeah. Okay. Thanks for talking about your rotifers to us. Yeah. So basically, main take-home message when using GenomeScope is properly check that your model actually explains the data, because sometimes you can get weird things. So now we have a little bit of another practical bit. So we're just gonna, we will start by fitting some haploid models. So just the histograms were generated with E. coli or Lambda. I think we also had some data from Lambda. You can start with those and using GenomeScope. I'm looking at the pretty graphs there. But you can also fit more data from this directory here. I'll copy it on the chat. And then we have a number of species in the directory that can be used. So basically we have 8 breakout rooms. So depending on the breakout rooms you get assigned, you can work with one specific species. And what we want you from this experience as well is that to sort of discuss with everyone else in your breakout room as soon as you get your model is, well, basically what characteristics are on your genome. Probably some of them will be more difficult than others, some can be trickier, but hopefully we'll leave some time for that and then chat a little bit about some of them afterwards. Does that sound okay? Did I miss anything?
So, I would encourage everybody to try multiple of those histograms, because fitting a Kmer model doesn't take that long. So let's now have 20 minutes of you fitting whatever you want, but and then have a 10 minutes where we share the histogram that is assigned to your group. So if you will be breakout room 1, let's find among yourself one peer that will just show us a begonia plot. Then if you breakout room 2 will have to show us the bambina plot, etc. Is that all right?
We have a raised hand, I think.
Can you ask the question then?
Yes. So in the web server, now it's clear to me after Lucia's presentation, but in the web server, when you have a field for maximum Kmer coverage, so actually that means, if I'm understanding well, the coverage of your sequencing. So related to what Lucia was saying, checking be careful about the model and the parameters, right? Is that I'm correct or I'm wrong?
Basically, wait, let me correct me if I'm wrong, Kamil, but basically what you're telling there is where how much of the coverage your model will take. So do you want these Kmers to be in your model or do you not want them? So if you want, you tell the maximum to be over here. But if you don't, you tell it to be here. And that's going to affect the genome size estimation, for example, and the repetitive content. Kamil is going to actually talk about that later with one very illustrative example that he has, but it's a very good question.
We'll talk about repetitive Kmers later on.
I also have kind of a question or maybe comment related to your previous example of the rotifera.
Yeah.
Could it be that the difference between the genome size that you estimated with the Kmers and the flow cytometry results is due to the repetitive contents, or like the amount of repetition in the genome. Because with the flow cytometry, you're kind of measuring how much of the whole mass is there. And with the Kmers, you're counting like how many different patterns you find. Is that possible?
So the Kmers take into account the repetitiveness. So basically, if the genomes are extremely repetitive, we're still able to very precisely estimate the size, although people sometimes stop counting after certain coverage. So no, this is again something we will talk about later. But the thing is, it could potentially expand part of it if it would be other way around. If you would have flow cytometry that says that the genomes are big and Kmer spectra that says the genomes are small. But if it's other way around, it's impossible. And we have one more question. Is there a particular reason why we use privilege K equal to 21? It's just a sweet spot. It's just a sweet spot where you have already the high complexity and it's low enough for you to have decent coverage.
Thanks.
But there is no particular reason for it. There are people that are like doing mostly 31 Kmers, people that are doing 27 Kmers. 21 Kmer is just my Kmer.